The increased growth rates of algae, including Pediastrum boryanum, in the Chesapeake Bay have caused blockage of sunlight and the usage of oxygen to the detriment of the bay's aquatic and ecologic health. The algae block sunlight, killing organisms below the surface, as well as using oxygen during growth and decomposition. Together, these factors cause harmful dead zones in the Chesapeake Bay and other bodies of water, such as the Gulf of Mexico. Algae growth occurs as a function of nutrient concentrations in waterways. Much of the nitrogen used on farms runs into the Chesapeake Bay. The purpose of this project was to determine if, and to what degree, nitrogen affects the growth of Pediastrum boryanum. In order to test our hypothesis that increased levels of nitrogen would stimulate higher algal growth, samples of Pediastrum boryanum were placed in solutions containing varying levels of nitrogen fertilizer. They were left under constant artificial light for 6 full days. On the 7th day, qualitative and quantitative measurements were taken for all 40 beakers of algae. The results showed that increasing nitrogen almost proportionately increases the growth of Pediastrum boryanum. The dry masses of the algae ranged from 4 grams to 12 grams, while the measurements of surface coverage corresponded to the masses. These results show the need for an effective policy limiting nutrient runoff into the Chesapeake Bay. Because much of the runoff comes from agricultural sources, it is important for farmers to limit nitrogen use. When it is essential for nitrogen to be amended to the soil, however, steps, such as terracing fields or digging ditches to reroute runoff, can be taken to prevent the nitrogen from traveling from the farms to the bay. In summation, it is vital for the health of our nation's water resources that we limit nitrogen runoff in order to decrease the dangerous growth of Pediastrum boryanum.